So, good evening, everyone. Welcome to the NPTEL Immunology lecture series of the live sessions. And today is week 9. We will be talking about complement system today. And I am Seemanti, a Ph.D. PMRF scholar in the Department of Biotechnology at IIT Madras. So I am a teaching assistant of this course along with Suraj Shripal. So today we will be discussing about the complement proteins or the complement pathway. Good evening, Shashwata. So what are complement proteins? We have frequently heard about this term, right? Complement proteins or the complement pathways. In our previous classes, we were talking about that these complement proteins play an important role in, phagocytosis, in killing the pathogen. So what are these complement proteins? So these complement proteins, these are set of serum proteins. They are present in your blood serum, and they play a major role in the effector pathways. What are the effector pathways generally? Effector pathways can include, phagocytosis or inflammation or uh, some kind of, chemical signaling pathways, say like chemokine signaling pathways. So that sort. Okay. So these proteins, they play a very important role in all these effector pathways associated with innate and adaptive immune systems, both innate and adaptive immune system, and they help in eliminating pathogen from the body. So basically, complement proteins initially that they are present inside the body or in the circulation, they are in the inactive state, and in the inactive state they are not having any kind of function as such like all these effector pathways, but they get activated and then they play a major role in all these effector pathways. We will see how they are activated and what are the different ways in which the complement proteins get activated. So before that, complement protein, the name or the term complement was coined by Paul Ehrlich. We have heard about this scientist earlier. He's a very famous scientist in the field of immunology. Ehrlich had discovered this magic bullet or magic shots, so Paul Ehrlich, he coined the term the complement protein, and he defined it as the activity of blood serum that completes the action of antibody. So what does this particular definition mean? So the activity of blood serum that completes the action of antibody, this definition came from certain experiments which were performed earlier by other scientists. So what they did is they had used the serum component of the blood, and they used a heat inactivated serum component, and they checked what would be the effect of that heat inactivated serum component, heat inactivated uh, serum component on the uh, elimination of general serum on the elimination of pathogen. So they wanted some earlier scientists, they had done certain experiments and they wanted to check the effect of heat inactivated serum on the elimination of pathogen. So when they heat inactivated the serum, so serum contains antibodies. So antibodies are proteins and they get denatured by heat or they are rendered inactive by heat. So when they used heat inactivated serum to incubated it with a pathogen, they could see that the pathogen is not eliminated. But again, what they did is they combined a heat inactivated serum with a heat not inactivated serum, like the normal serum, and they again checked the effect of that combined heat inactivated plus normal serum on the elimination of pathogen. So they could see that when they are combining both of them, the pathogen gets eliminated. So what happens is there must be something which is present in the serum component that is, complementing for the activity of antibody. So that complementation function is mediated by this protein, which are known as complement proteins. So complement proteins, generally they are working along with the antibodies in order to in order to carry out opsonization or carry out the phagocytosis of the pathogens. So so complement proteins, they are very important part of both the innate and adaptive immune systems. They are serum, they are major component of the serum proteins, and they are essentially they complement the role of antibody in the body. And they are generally present in the inactive state in the blood. So there are this inactive, generally they are not in the active state, and they are not performing any kind of these functions. Once they are activated, then they will carry out all of these effector functions. So there are three major pathways that lead to the activation or cleavage of these inactive complement proteins. And once they are activated, there is a cascade of activation that takes place. So one complement protein is going to activate another complement protein. So complement proteins are named as C1, C2, C3, etc. And so the cascade of complement proteins gets activated, and then this complement proteins ultimately results in the formation of a membrane attack complex that we know that we denote as MAC. And this MAC complex is that this MAC complex is a cylindrical pore-like structure. It is present on the surface of pathogen. So your pathogen has a cylindrical pore-like structure, and this cylindrical pore-like structure is called MAC, which results in the osmotic imbalance in the pathogens, and that ultimately causes the water or fluids to enter this pathogen. Here pathogen, the water or fluids will enter, and that will cause swelling up of the pathogen. The pathogen will get swelled up, and finally it will burst. So the formation of membrane attack complex is the significant. It shows that the cells are going to die. And MAC complex is a very it forms a very big pore. It's around 70 to 100 angstrom diameter, and it's a very big pore, and it definitely leads to a very deadly lysis of the pathogens. So complement proteins, what are the different proteins which are involved in the complement system? So first we'll see that complement proteins, their nomenclature is C1, C2, C3, C4, and so on. It goes on till C9. So we have C1, C2, C3, C4, C5, C6, C7, C8, and C9 proteins. And so these complement proteins, these are like whole proteins. They have when they are cleaved, so these are inactive proteins. These are present in your circulating blood. Once they are cleaved, they will be having generally two subunits, which are named as C2A or C2B, for example. If C1 is cleaved into two, then it can be C1A or C1B. If C3 is cleaved into two, it is C3A and C3B, and so on. So generally, all these complement proteins, they get cleaved into this two subunits, and of that, one is a smaller subunit and the other is a bigger subunit, and one is membrane bound while the other is soluble. So generally, some fragments, for example, in most of the complement proteins, the B part is membrane bound part. Most of the complement proteins, in case of C2, it is the opposite. So yeah, membrane bound part is mostly the B, and soluble portion is mostly the A part, with some exceptions like C2. Uh, and the smaller fragments are denoted as A, while the larger fragments are denoted as B. So this is what our complement proteins nomenclature is all about. So how is complement system working, and how are these different proteins recognizing the pathogens, and how is the membrane attack complex formed, how is the inflammation carried out, how is the phagocytosis carried out? So we'll look into each of these concepts. So initially, what happens, what are the initiators of the complement system? So initiators of the complement system are this basically proteins that are directly or indirectly binding to your pathogen. So proteins that directly or indirectly interact with your pathogen. With pathogen. So, what do you mean by directly and indirectly interact? So, directly interact are some proteins like MBL or phycolins. These are MBL stands for mannose binding lectin. So MBL and phycolins, these are present on the surface of your. These are present on your in your blood. Normally, these proteins are present in your blood, and they can directly interact with the antigens present on your present in the circulating system. So some proteins like MBL and phycolins, they can directly interact with pathogens. So they do not require any like antibody antigen interaction to recognize the pathogen. They can directly interact with the antigenic epitope of the pathogen. So these are MBL and phycolins. Whereas some proteins, they require some proteins of the complement pathway like C1q. So C1q is one of the subunits of the complement protein C1. So one of the subunit of complement protein C1, and C1q requires that the pathogen or the antigen is recognized by the antibody. So this is the antibody, green colored portion is the antibody, this red color portion is the antigen, and this portion that you are seeing here, this is C1q. So C1q recognizes the Fc portion of your antibody. This is the Fc portion of your antibody. Okay, so once the antibody is bound to the antigen, the Fc portion of this antibody undergoes certain conformational changes, and these conformational changes will allow the activation and binding of the complement protein C1. So there are other subunits of C1 as well, that is C1R2, C1S2. So those are activated after C1q binds to the Fc region of the antibody. So initiators belong to these three kind of proteins generally. One is the C1q, second is MBL or mannose binding lectin, and the third is phycolin. So these are the initiator proteins. So once the antigen is recognized by the antibody, or it is recognized directly by the proteins, complement proteins, or it is directly recognized by the pathogen associate, like the pathogen recognizing proteins. So once it is recognized, what will happen is there will be a cascade of events that will lead to the formation of mainly two enzymes, that is C3 convertase and C5 convertase. So C3 convertase breaks down C3 into C3A and C3B, whereas C5 convertase breaks down C5 into C5A and C5B. Now, once your initiator pathways has started, so your C1q or MBL or phycolin recognizes your pathogen, and a cascade of events will follow that will lead to the breaking down or cleavage of several other proteins like C1R, C1S, as I was talking about different subunits of complement protein C1. Then C4 protein gets broken down into C4A and C4B. C2 protein gets broken down into C2A, C2B, and likewise. So once it is broken down, and we'll look into the details, then we'll see that some enzymes are formed, that is C3 convertase and C5 convertase, that results in the breaking down of these two complement proteins, important complement proteins C3 and C5. Why are these important? Because C3 protein, from like once C3 protein gets broken down, then only we can proceed towards the C5 breakdown. And C5 breakdown is important because if it is not broken down, then we will not be able to form the membrane attack complex. So all the pathways for activating complement system initially starts with activating the C3 convertase, that ultimately leads to the activation of C5 convertase, and finally the formation of the MAC complex. So once it is once the initiator pathway starts, initiator protein starts the activation of complement, then several proteins gets broken down, several complement proteins get broken down. So see, this is suppose is a resemblance of a complement protein. Okay, so say C2, and it will get broken down into C2A and C2B. So one is the red portion and the other is the blue portion. So one will be membrane bound and the other is free or soluble form. The one that is soluble, whichever fragment is the soluble portion, that soluble portion is generally associated with the anaphylotoxins or inflammation. So anaphylotoxin or inflammation, all the soluble forms of the complement proteins which are broken down, the soluble parts, they run in the circulation and they activate the inflammatory pathways. They will activate the cytokines and chemokines to be released, and that will result in extravasation. So what do you mean by extravasation again? So if there is any kind of wound over this site and your complement proteins are circulating towards your in your blood, and then it is going and activating the inflammatory pathway, so your cytokines and chemokines are released over here. So all this immune cells, WBCs, neutrophils, monocytes, etc., they will be pulled from the circulation towards that site of the wound. So this process, extravasation, is the process by which this immune cells squeeze out from this epithelial boundary of your blood vessels to go and reach this site of wound. So that function is mediated by the soluble fraction of the complement protein that is cleaved. So what happens to the membrane bound one? So the membrane bound one can have two different functions. One is the membrane bound one is binding to this antigen surface. So this is, say, the red dots. These are the red dots which are shown over here. These red dots. These red dots are antigen. Antigen are bound with the antibody, and on the top of this antibody you see these blue colored small dots. These are complement proteins, membrane bound fraction. So this membrane bound fraction, they go and bind to this Fc region of this antibody, which is already interacting with your antigen, and they coat the surface of your antigen. So your antigen, it is coated with antibody and uh, complement protein. So these are complement protein, and these are coated on its surface with you know antibodies, sorry, complement proteins, and these are antibodies which are recognizing all your antigen. So the antigenic surface is coated with antibody and complements. So antibody plus complement proteins on the surface of your pathogen is going to be recognized by phagocytic cells. What are phagocytic cells again? Phagocytic cells are macrophages, they can be dendritic cells, they are neutrophils. So all these phagocytic cells, they will go and bind to this, not dendritic cells, okay. So phagocytic cells are mostly macrophages, monocytes, etc., neutrophils. So so they will go and bind to this antibody and this complement proteins through their complement receptors. So phagocytes, in their surface, they have complement receptors. So this blue portion that you are seeing over here, these are complement receptors present on the surface of your phagocytic cell. This is the phagocytic cell. So phagocytic cell has complement receptors. These complement receptors binds to the complement proteins that are already bound to your antibody. So you are seeing this blue colored antibody. On top of that, you have this small light blue colored complement protein, the membrane bound fraction of the complement protein that is bound by the complement receptors on the phagocytic cells. So what it does is it helps in taking up this antigen more quickly, or it helps in phagocytic activity of this cells of this antigen gets very efficient. Like this antigen will be very quickly phagocytosed by this cells if complement proteins are also coated on the surface along with an antibody. So what are opsonins? So these are called as opsonins. Whatever like antigenic surface proteins, if some proteins, if they are coating the surface of antigens, and those proteins, if they help in taking up this antigen by the phagocytic cells more efficiently, then they are known as opsonins, and the process is known as opsonization. So opsonins are any proteins or any molecules that when coated on the surface of pathogen helps in increased phagocytosis of the pathogen. Right. Hmm. And the process is known as opsonization. The coating of opsonin on the surface of your pathogens is known as opsonization. So one of the function of complement protein is to act as an opsonin. So it will coat the surface of an antigen and help in taking up this antigen by the phagocytic cells. The second function is as an anaphylotoxin. What is anaphylotoxin again? Any toxins which can cause anaphylaxis. Anaphylaxis is a kind of inflammatory shock kind of thing. Inflammation, a huge inflammatory response is known as anaphylaxis, and so it can act as an anaphylotoxin, or it can be involved in inflammation, to be in, more in general terms. So the soluble portion of your, the complement protein is associated with anaphylotoxis, whereas the membrane bound is associated as an opsonin or a membrane attack complex. What happens in the membrane attack complex again? So the membrane bound form of this complement proteins, once it gets started, the activation gets started, and there is a cascade of C2 gets activated into C2A, C2B, then it will get activated into C3A, C3B, then it will get activated into C4A, C4B, then gets activated into C5A, C5B. So from here again, you see the C5B will form a complex with C6, C7, C8, C9, C9, C9. So there will be a lot of C9 monomers that will get uh, inserted into the membrane of your pathogen, and this membrane will get a cylindrical shape due to the insertion of multiple multimeric C9 molecules, and this is known as the MAC complex, which is responsible for the lysis of the cell. So this is the all membrane bound form. So these are forming complex, and finally a MAC complex is formed over here, that is known as the membrane attack complex, and this results in the lysis of the cell. So we have seen that there are three important functions: as an inflammatory response, for lysis, as well as for opsonization. Also, another thing is that in complement system, another important protein that plays a role is, so if all the time your complement proteins are active, then your MAC will be formed in most of the cells, or it can lead to non-specific. It happens that sometimes non-specific, your MAC complex is formed. But to degrade it, we can have this complement protein, some regulatory proteins that can degrade these complement components. So this yeah so this complement proteins have some kind of other regulatory proteins that can bind to these complement proteins and result in the inhibition of formation of MAC complex and deposition of this component. So all this C2, C3, this will not get cleaved, or the cascade will not follow whenever this regulatory protein is binding to any of the complement proteins in the initial stages. So if it is binding to C3b, then C5 convertase will not form. If it is binding to C4b, then C3 convertase is not going to form. So all of these initial pathway proteins, if they are bound to some kind of regulatory proteins, they will prevent the deposition. So it will act as an umbrella or a shield to protect this cell from getting deposited by any complements. So these are the regulatory proteins that are again another part of the complement system. So what are the three What are the three pathways for complement activation? Again, the three pathways for complement activation are the classical pathway, the lectin pathway, and the alternative pathway. So, of which these two pathways, they are mostly antigen antibody interaction dependent. And alternative pathway is antibody independent pathway. Uh, hmm. So let us go through briefly what is each of this pathway and how does it activate the complement proteins. So as I said, complement proteins are C1, C2, C3, C4 and goes on to C9. So what happens in the classical pathways? So antigen is recognized by your antibody and the Fc region of your antibody. So this is the Fc region of your antibody. This Fc region of the antibody will undergo certain kind of conformational changes and that will allow the binding of circulating protein C1 and of C1 proteins the subunit is Q. So C1q recognizes Fc portion of the antibody which is already bound to your antigen and this will bind to this antigen antibody complex. Once it is bound to the antigen antibody complex, the C1q gets activated and it will go and bind and activate the two other subunits of the C1 protein, that is R2 and S2. So together what will happen, that together we can call it as C1q R2S2 activated. So this dash over here signifies that this R2S2 portion is activated or cleaved. If it is not cleaved, then we will not use the dashed form on the top of R2 and S2. So once C1q recognizes the Fc region of the antibody that is already bound to an antigen, it will go and activate the R2 and S2 fragment and so C1 R2 S2 gets activated with due to the binding of C1q and that will result in the cleavage of two complement proteins in the pathway, that is C2 and C4. So C2 gets cleaved into C2A and C2B and C4 gets cleaved into again C4A and C4B. This is C4B. So this is what happens in the classical pathway. Once it is bound to the C1q R2S2, it will cleave C2 and C4 complement proteins. This will get cleaved, C2 gets cleaved into C2A and C2B, C4 gets cleaved into C4A and C4B. Now when C4B and 2A, so C4B and C2A, they form a complex and this complex is known as C3 convertase. So the main goal for all of this pathway is to come and merge towards forming the C3 convertase. So all three pathways will merge together and form the C3 convertase uh, in different ways. So each of these pathways will form ultimately the same thing but in different ways. So lectin pathway, what it does is, instead of antigen antibody interaction binding to this antigen, lectin pathway will result in the binding of the carbohydrate moieties which are present on your pathogenic surface. So these are carbohydrate moieties. These are directly recognized by certain proteins which are known as mannose binding lectin or phycolin. Okay, so these proteins directly recognize these carbohydrate structures present on the surface of pathogen, and recognizing this carbohydrate structure does not require antigen antibody interaction directly, but again it is why it is known as antigen antibody interaction dependent pathways, because antigen is interacting with some kind of protein, not necessarily it is interacting with antibody at this point for activation of complement, like when we are looking only the complement activation per se, but it is somehow it is associated with the antigen recognition. So that is why it is known as this antigen antibody interaction dependent. Whereas in alternative pathway, we do not require any kind of antigen antibody interaction to activate the complements over here. So that is why here this classical pathway and lectin pathway is associated with this antigen interaction or antibody release and antigen antibody interaction. So what happens in the lectin pathway is, again, the carbohydrate moieties, they are recognized by MBL or phycolin, and this will result in the, again, conformational changes over here in these proteins MBL and phycolin, and these proteins which you are seeing over here, these proteins are known as MASP. So MASP, these are serine proteases. So SP stands for serine proteases. So these serine proteases are responsible for cleaving again the C2 and C4, and this gets cleaved again into C2A and C2B. C4 gets cleaved into C4A and C4B, and finally again we are finding the formation of the C4B combines with C2A to form C4B2A, that is C3 convertase. So this is what lectin and classical pathway ultimately converges to. What happens in the alternative pathway is there is spontaneous hydrolysis of C3 protein. Okay, irrespective of whether there is an antigen antibody interaction, or whether antigen is recognized by some other immune cell or some other protein like MBL or phycolin, C3 will undergo spontaneous hydrolysis, and this spontaneous hydrolysis will result in the formation of a subunit like C3H2O-BB. This particular component, this particular fragment of C3 after spontaneous hydrolysis can bind to some other proteins like factor B or factor D, and get converted to C3B and C3A, along with this factor B small portion, small fragment, that is BB, capital B small b, means factor B's factor B also has a BA or BB, and so once it gets hydrolyzed into, initially when it gets spontaneously hydrolyzed into this fragment, the other hydrolysis can take place where C3 is getting broken down to C3A and C3B. So there are two hydrolysis reaction that is taking place. One is resulting in the formation of this fragment of C3, and the other is resulting in the formation of these two fragments of C3. So C3B will go and bind with this C3 C3BB will bind with C3B and form C3B capital B small b. So this is happening after interaction with factor B, D and properdin. So these proteins are necessary for keeping all this complex together. So factor B binds to C3B, and on top of that there is factor D, and all this complex is all these proteins are required to keep this complex together, and this results in the formation of C3B capital B small b. So this C3B capital B small b is also a C3 convertase. So C3B capital B small b is a C3 convertase. Then C4B2A is also a C3B C3 convertase. So this two C3 convertase again breaks down C3 into C3A and C3B. So here also see you are breaking down C3 spontaneously into C3 and C3B, and again we are converting that into C3 convertase to break down more amount of C3, because we need complement proteins to be activated in more amounts, in huge amount, because we need to coat the entire surface of your pathogen, and there is not just one pathogen, there is so many pathogen in your body. So you require a lot of C3 proteins in your circulation, activated C3 proteins, C3B proteins in your circulation. So your C3 proteins get activated, C3 convertase proteins get activated, and they cleave C3 into C3A and C3B, of which this C3B will go and bind to the already existing complex of C3B capital B small b to form C3B capital B small b and C3B again. and this complex has C4B2A, again a 3B will add over here to form, so here C3B is added, here also one C3B is added. So we will see that it is forming, this is forming C3B, capital B small b, again 3B, and this is forming C4B2A 3B. So these complexes that are formed after addition of C3B, this is resulting in the formation of a C5 convertase, and this C5 convertase will break down C5 into C5A and C5B, where C5A is associated with inflammation and C5B is associated with lysis. So C5A is again the soluble portion of your complement protein, and this is the membrane bound portion. So likewise, all these complement proteins, they are broken down into a soluble fraction and a membrane bound fraction. Soluble fraction is associated with inflammation, membrane bound fraction is associated with lysis or opsonization. So again over here also we can see that C3B, this is the membrane bound portion, it is associated with opsonization, whereas the soluble portion, the C3A, C4A, associated with inflammation. So you see most of the proteins, the A subunit is associated with inflammation, exception is C2A. So C2A is the membrane bound form, this is an exception. Rest most of the complement proteins, this the A subunit is the soluble form, okay, and this is associated with inflammation. So what are the other events that are leading to the formation of C5 convertase? Again in some details where some text format where it is written more clearly. So C1q, it binds to the antigen bound antibody. This is the C1q, this green color shape like structure, this green colored everything. This is the C1q subunit of your complement protein C1. They will bind to the antigen antibody complex. So antibody binding sites on the antigen is this. Antigen and antibody interaction is taking place. So conformational change in the Fc region of the antibody, that will result in the binding of C1q subunit of your C1 protein, and this will induce a conformational change in one of the C1R molecule. So this portion of the complex is the C1R2S2. This results in the conformational. So once it is bound over here, here also some conformational changes that take place, and that results in the activation of C1R2S2 subunit. So C1R then activates the second C1R and the two C1S molecules. So C1q binding will result in the activation of C1R2S2. Then what happens is C1s will cleave C4 and C2. C4 is cleaved first and C4 gets cleaved into C4a and C4b. C4b will go and bind to the membrane. So C4b is over here. So it is binding to the membrane. And C4b will go, so once C4b is binding to the membrane, there will be another complement protein that is cleaved by this complex, that is C2. So C2 will go and bind to the C4b. So C4b will bound to C2 protein, and C2 protein will then get cleaved into C2a and C2b, of which the C2a portion, that is already membrane bound, it will remain attached to the C4b portion of the complement protein, and the C2b will be released in the circulation and act as an inflammatory agent. You understood? So C4 gets cleaved by C1 into C4a and C4b. C4a is released into the circulation. This is into the circulation, and C4b gets embedded in the membrane. Then this C4b over here, some other protein, that is complement protein 2, C2 protein gets and binds over here. So this is C4b bound to C2 protein. Here the C4b protein will cleave C2 protein into C2a and C2b, where C2a will remain. So this is getting cleaved into half. C2a remains bound to the C4b, and the C2b gets again released into the circulation. This C4b C2a complex is known as the C3 convertase. Now, once the C3 convertase is formed, C3 convertase will hydrolyze many C3 molecules. So C3 molecules will be hydrolyzed into C3b and C3a. So of which this C3b will again come and attach over here and form C4b, 2a, 3b. This is the C5 convertase, and this C5 convertase will again bind to C5, where C5a and C5b will be formed, and again C5b will be attached over here. So your total self will be C4b, 2a, 3b, 5b. So this is what the C5 convert. So this is what your uh, complex will go on forming, and it will ultimately result in the formation of a MAC complex. So this is the events that lead to the formation of C5 convertase, and formation of C5 convertase. Once C5 convertase is formed, it is self-sufficient enough to call all other proteins and form this MAC complex and carry on with the lysis. So how is all these proteins bound to the membrane? So basically, you have seen that these proteins, these are uh, these proteins are bound to the membrane, and how are they bound? Why are they bound? Basically, so these proteins, both all this membrane, membrane bound portion of this protein C4b, C3b. So all this membrane bound portions, they have a very highly reactive thioester bond. So this is a thioester bond that is present, and this thioester bond can easily be cleaved over here. How they can be cleaved? By this NH2 amino terminal or OH group if it is present in your cell membrane. So these bacterial cell membranes, they have OH group, and they have mostly pathogenic membrane, they have this NAM-NAC protein, this NAM-NAC complexes, or they have a lot of carbohydrates, lot of proteins on their cell membrane. So these proteins and carbohydrates, they can easily attack this thioester bond and cleave this thioester bond. And so basically those cell membrane of those pathogen are meant to protect them, but they can also be harmful for them in some cases, like here. So they are, those proteins and those carbohydrate moieties, they can easily attack this bond, and because this is a very highly susceptible bond, and it can undergo a nucleophilic attack by all these side groups of the proteins and carbohydrate moieties, and this will result in the cleavage of this thioester bond. And once this thioester bond is cleaved, so this bond will be this bond is broken down. So this bond is broken down, and your cell membranes, this amino terminal gets linked to your 4b protein molecule. So C4b protein molecule gets linked to your bacterial or your pathogenic cell membrane. If it is not linked, or if just the hydrolysis reaction has taken place, so it could be just the COH has formed, and here the bond has been broken down, but there has not been any linkage. This is very prone to linkage, but yet there sometimes they do not show, because they might not be in very close vicinity to create that attack. So this is the different ways. So this is the different situations in where C4b is in the unbound state, or in the bound state, or in the hydrolyzed state in the surface of the pathogenic cell membrane. So here you see that C4b and C3b, they have highly reactive thioester linkages, which are susceptible to nucleophilic attack by cell membrane proteins or carbohydrates. So formation of covalent bond between membrane macromolecules and complement components. So this is a covalent bond that is formed. So there is no non-covalent linkages between the cell membrane and the membrane bound portion of the complement protein. So all these are covalent linkages, and they are very strong, and therefore they coat the surface on the proteins in very strong manner, and they are not easily removed. So so how is again the formation of MAC complex carried out? So yeah, so once you know the C5, once the C5 convertase is formed, so C5 then gets broken down into C5a and C5b. then the C5b gets embedded in, it's already embedded in the membrane, right? All these membrane bound components are embedded. So C5b will bind to another complement protein and help in embedding this C6 complement protein in the membrane. Then it will go and bind to, this complex will go and bind to C7, this will go and bind to C8, and then C9. There will be multiple monomeric units of C9, and this will form a huge pore in the cell membrane of the bacterial or the viral or whatever pathogen. And those membrane, from here, the cell, water might come out or water will enter, and this will cause lysis of the cells. So here one important term is innocent bystander lysis. So what do you mean by innocent bystander? So any kind of cells that are present in the vicinity of your target cells, they are just standing there, they are innocent bystanders. So they are just standing there, they are not having any role in causing any infection to your body. But sometimes your MAC complex or your accumulation of your MAC, not even the MAC complex, before that, so just C5b67, so this structure basically C5b67, if they are accumulating on the surface of a normal cell, a normal your own self cell, so that cell will get killed. So that is an innocent cell that will get killed and lysed instead of the target cells. So it will happen because C567 is self sufficient to get deposited on cell membrane surface and form pores on the surface. So C6C7, once if it is deposited on the surface, it's going to form the MAC complex, and finally it is going to form a pore and get the innocent cell killed. So sometimes it happens that not your target cell, but all the cells which are lying in the vicinity of your target cells get killed. So that is known as innocent bystander lysis. Another important function for complement protein is the anaphylatoxin or inflammatory response. So all these soluble portions, C3a, C5a, these are the soluble fraction of your complement proteins. They will go and bind to the complement receptors. So these are complement receptors. So C3a binds to complement 3a receptor. So these are complement receptors basically. These are complement receptors. So all these complement receptors bind to all your soluble portion of your complement proteins, as well as the membrane bound portion when they are carrying out the opsonization pathway that I said in the first slide. So here these complement receptors, first slide again, just so these complement receptors present on the surface of phagocytic cells, they help in opsonization. Okay, and these complement receptors which are present on the surface of your normal cell, phagocytic cell or whatever cell, here also it helps in signaling pathway. How it is helping in the signaling pathway? So these complement receptors, they are associated with another protein known as GPCR, or G protein coupled receptor. So G protein coupled receptor, it is a very important receptor class of family of receptor proteins that are associated with several cell signaling. So G protein coupled receptors, they help in activation of CAMP, they help in activation of protein kinase A, and they help in activation of several transcription factors and this and that. So this complement proteins, they will bind to this complement receptors and they will activate this GPCR protein. So GPCR has three subunits, alpha, beta, and gamma. And so in its inactive state, GDP is bound to the alpha subunit. Once GPCR gets activated, GDP gets replaced by GTP. So this alpha subunit has now GTP. Now this is activated. This will go and activate protein kinase A, and this will go and activate the several cytokine signaling molecules. Cytokines will be released. So all these genes which are produced, which are associated with cytokines release, will be activated. So so through the GPCR signaling, the inflammatory response or the anaphylotoxis response is initiated, And in different cell types, the different kind of cytokine mediated signaling is taking place. So for example, in macrophage, cytokine production, phagocytosis, chemotaxis, everything is associated with these complement proteins. In neutrophil, chemotaxis, oxidative burst. Oxidative burst is in like in presence of oxygen, the cells will be dying. So it will help in killing the oxygen mediated killing of cells. Basophil, it helps in degranulation. What do you mean by degranulation? So any kind of cells which are having this histamine, so these are histamine and all these prostaglandins. These are pain mediators. So they help in releasing all of these histamine molecules from this basophil, and this will help in alerting the other cells in the vicinity to be careful enough that there is a protein, there is a infection in your body. So likewise, eosinophil is also associated with degranulation and chemotaxis. Mast cell is associated with degranulation and chemotaxis. So different cells are associated with different effectors of effector functions whenever a complement protein is binding to their complement receptors. So complement receptors present over here, and this complement receptor is bound to complement protein. This complement protein will help in all of these functions of each of these cells. And these complement receptors which are present on the surface of the phagocytic cells. So all of these are complement proteins, so C1q, MBL, mannose, antibody antigen, C3b, C4b. C3b cleavage products are different kinds of C3b cleavage products are also there. So it's not just C3a and C3b. C3b gets cleaved into these several subunits, and these subunits can be regulated by different kind of proteins. And so that will not go into details, but just know that C3b has not just it's not just stopped over here. C3b can also be cleaved into C3b, C3c, and C3dg. So all these proteins, these are present on the surface of your antigenic cell, and they will be recognized by the phagocytic cells having complement receptors. And these complement receptors are CR1, CR3, CR4, CR1G, FC receptors, SINGR. So these are the names of all these complement receptors which are recognizing all of these proteins. And these are some memory based stuff, like what kind of ligand does it interact with. So it interacts with CD35, this interacts with CD11b, CD18. So these are something that you might remember, that what complement receptors interact with what kind of ligands. And finally, what are the functions of complement protein? So mainly there are 4 functions. So lysis, so it will form a MAC complex on the surface of pathogen, and they kill it by osmotic imbalance. Opsonization, so it will act as opsonin. Again, I explained what was opsonin. So it helps in coating the surface of pathogen along with the antibody molecules, which will render the pathogen more susceptible to phagocytosis. Inflammatory response, so C3a and C5a, these are majorly associated with enhancing the inflammation and anaphylotoxis. And chemoattractants, it also directs the entry of immune cells through extravasation to the site of infection by release of chemokines. So complement proteins coating the surface of an antigen will help in releasing all these inflammatory chemokines and cytokines over here. Thus, like over all of these cells we release any kind of chemokines and cytokines based on whatever cell type it is interacting with, and that is going to call or direct more cells towards the site of infection. So these are some of the regulators of complement proteins. You can go through that. There are just it's just given the name as well as the function and which pathway they are affecting. So C1 inhibitor protein, it will affect the classical and lectin pathway, obviously, because alternative pathway starts from C3 protein. So C1 inhibitor will bind to C1 and not allow the dissociation of C1R2S2 from C1Q. So C2 and C4 will not get cleaved. Again, this decay accelerating factor, this is affecting all the 3 pathways, classical, alternative, and lectin. So it will accelerate the dissociation of C4b2a and C3b-BB. So it will bind to the C3 convertase, and it will, the complex of C4b2a will be dissociated, or C3b-BB will be dissociated, and thus the C3 convertase is not formed. Likewise, CR1, this is again a complement receptor. This is also involved in regulation So this, it blocks the formation or association of C3 convertases by binding to either C4b or C3b. So likewise, all of these proteins, they are associated with different function and regulation of the complement activation. So now we will just go through some questions. So you can basically try to solve this. That's right. So the answer is option D. All of these are the functions of complement system: lysis, opsonization, inflammation. So all of this is what complement system is. Complement system does. So next question. Right. So the correct answer is option A. Complement protein C1 binds to Fc region of all this antibody and antibody antigen complex, and then that associates with different conformational changes, and again the activation of the complement cascade. The third question. No, Rajkumar, why will it be option D? Yeah, so the correct answer is option number B. Kirtana, it is not C4 convertase. We have talked about only two kind of convertases, that is C3 and C5. C4 convertase is not a very like important for our scope of study. So C4B2A is associated with C3 convertase. It is associated with breaking down of C3 protein into C3A and C3B. So it's a C3 convertase. The answer is option number B. Okay, so whatever the so you are seeing, so this is a smaller name, so it's C3 convertase. If you are getting another small B over here, 3B over here, or a bigger name, that would be associated with C5 convertase. Okay, so yeah, so the next question. Right. So everybody, you have given a right answer. Answer is option B. innocent bystander lysis is a phenomena where this complement protein, it will kill all these nearby target cells instead of the target cell itself, and it's going to kill the innocent cell. So it is known as innocent bystander, the term. So that's right. The last question for today, and then we'll wrap up. Pooja, it's not just option A. See, we have the both option A and B has the A fragments, smaller fragments, C3A and C5A. So all this soluble portion mainly are A subunits or the A fragments, as I said, except C2's protein, C2A, that is membrane bound. So all of this A, this is associated with inflammation because these are soluble portion and they are going into the circulation. So the answer is both A and B, option number D. So both A and B is correct because both A and B is the smaller subunits C3A and C5A, and A is associated with inflammation and anaphylotoxis. Okay, so not just C3A, both C3A and C5A. So that's all for today. Uh, do you have any other questions?
Ma'am,
Yeah. Yes, Raj.
C3A, C5A,
Hmm.
smaller subunits are C3B are which kind of subunits, ma'am?
C3b is the larger subunit, and C3, all the A subunits are the smaller subunits. All the B subunits are the bigger subunits. Okay, there are two parts. See, one is
Yeah,
smaller
ma'am.
subunit and soluble, and one is larger subunit and
Okay.
insoluble or membrane bound.
Yeah.
So
Yes, ma'am.
C3b,
C3B is
membrane bound.
large subunits.
Yes, and membrane bound subunit.
Mm.
Hmm.
Okay, ma'am. Thank you, ma'am.
Okay, so that's all for today. Thank you, everyone. Have a nice weekend. We'll meet again in the next class. So we are already nearing.
Ma'am,
Yeah. Say, Rajkumar, you want to say something?
ma'am, exams will to October 29, ma'am.
I think so. You will get the information when the exam is in the the discussion forum. So you will get to know.
Ma'am, 1 second, ma'am.
Hmm.
Kirtana, Kirtana இருக்கீங்களா? So okay, ma'am. Thank you, ma'am.
Okay. Thank
Kirtana,
you. You have any question, Kirtana? Is Kirtana there or she left?
left.
Okay, okay, she left. Okay. Okay, then next class, maybe if you have any doubt, we'll discuss about that.
Okay, ma'am.
Okay. Okay.
Any another Tamil persons, ma'am?
Any Tamil person in the class now?
No.
No, I don't think so.
Okay, ma'am. Thank you, ma'am.
Okay, okay. Thank you. Thank you, everyone.